Swayam Prabha. Digital India. Educated India.
Let me welcome back again in my course, Industrial Biotechnology. In the last class, I tried to find out that what is the role of life cycle for the growth of the cells. I told you whenever we do the inoculation of the, because different in the growth cycle we have different phase, and different phase has some different significance. Lag phase is considered as the acclimatization phase, the growth phase is considered, the log phase is considered as the growth phase where the organisms are active, and in the stationary phase is considered as the starvation phase where most of the secondary metabolite formation take place. Now, when you do the inoculation of the cells, always we do the inoculation of the cells in the either mid log phase to late log phase, just to ensure the organisms are active. Now, we use the Monod equation. I showed you the Monod equation for the study the growth of the organism. So, it is similar to Michaelis-Menten equation, only we come across the S which is considered as the growth limiting substrate, and growth limiting substrate might be anything because media comprises of n number of chemicals. It may be carbon source, it may be nitrogen source, it may be vitamin, it may be minerals. So, anything might be the growth limiting substrate, provided the growth is if a mu, if it changes with respect to change of concentration of the particular component present in the reaction mixture, then and only then we call it growth limiting substrate. Then we come across the batch process and the chemostat process. Batch process is the process through which we can have this, we can find out the life cycle of the cells, but major disadvantage of the batch process is that we cannot hold particular phase of growth for infinite period of time. As the time passes on, one phase we switch over to the other phases. But in the chemostat process, we find that under sterile condition and steady state sterile feed and steady state condition, mu equal to D. So, by controlling dilution rate, it is possible to maintain a particular phase of growth in the fermentation broth. So, we can suppose particularly Baker's fermentation process, we need more cell mass production. So, naturally that rate of cell mass production will be more in the growth phase, but in case of batch process we cannot hold the phase for longer period of time, but in case of chemostat process we can continue this growth phase for infinite period of time. I think and how to find out, how to calculate this growth of the organism, how to calculate the substrate degradation, I showed you in the next class. Now this particular lecture I try to concentrate that we will come across the Dmax and D washout, which is very important as per chemostat operation is concerned. Then this can be overcome by using either by immobilization of the cells or by recycling the cell mass to the reactor, and how we can do that. So, this I am going to highlight in my lecture. Let me start with that. First, let me show you how you can calculate the Dmax. The Dmax is considered as the maximum, the dilution rate at which we will get the maximum rate of cell mass production. This is equal to, I showed you before that d into x is nothing but dx by dt. What is dx by dt? Is the rate of growth of the cells. Now, this will be like this, this will grow like this, and this will come down like this. Now, at the same time we can have the plot of x, xs is the steady state cell mass concentration, ss is the steady state substrate concentration. Now, steady state substrate concentration will be like this, it will go like this, then it will be constant, and then your cell mass concentration will come back here. That this is XSS and this is SSS. So this is very important because as your dilution rate increases, because this is the different dilution rate, this is D1, this is D2, this is D3. Now I told you dilution rate is equal to what? F by V. So in case of any kind of chemostat, suppose this is the chemostat. If we pass this is F and this is V. So we can change the value of D if you simply increase the value. V is constant. The volume of the reactor, volume of the working volume of the reactor is constant. So if you want to change the value of D, we can change the value of F. If F value change, D value will change. So at different dilution rate, and I told you that when you change the value of A after infinite time, you will get the steady state condition. What is steady state condition? When the cell mass condition will remain unaltered, or when the substrate concentration, cell mass concentration remain unchanged with respect to time, that is called steady state condition. So at different dilution rate, you will get the different steady state condition. So this is exactly what is happening, that as the dilution rate increases, your steady state cell mass concentration, and finally this is the crucial point where x equal to 0. X is equal to 0. No cell is present in the reactor, because if there is no cell present in the reactor, there will be no reactions, because cell mass is usually responsible for carrying out the reaction, and this is considered as a D washout. D washout means that at the dilution rate at which there is no cell present in the reactor, this is the D washout. And here you this the dx by dt, this is dx by dt with respect to time, it increases and then it decreases. Now here we have a point, and this point we consider as the Dmax. What is Dmax? Dmax is the dilution rate when dx by dt equal to maximum. dx by dt is the maximum. That means what we can write, that when this at this particular point, I can write at this particular point dx by dd, this should be equal to 0, because this is d, this is the dx, this is dx. So and dx by dd, this remain plateau. So I can so at Dmax, this is the condition that we have. Now let me come here, that here we can write that dx by dd, we can write like this, because we have previously we have seen that this is the expression that we have. Then S0 we can write like this, S0 by k we can write like this. Ultimately we will come this equation, and this equation is what? This is d equal to mu max 1 plus minus root over Ks plus S0. Now one thing I want to point out, that this is the Dmax value. This is the Dmax value. This we can calculate like this. Now the Dmax value always should be less than mu max value. So it cannot be plus, it will be always negative. So usually the Dmax value will be mu max 1 minus root over Ks plus, though it is in the we derive this plus minus, but actually the plus cannot be possible, it always should be less than mu max. So it is the minus root over Ks by S0. Now here the Dmax equal to like this, but since Dmax is much less than mu max, so we can write it Dmax equal to mu max 1 minus root over Ks by S0. Now that operation of the chemostat Dmax is the maximum rate of cell mass production from the reactor. The cell mass, now how we can find out what is the maximum rate of growth of the cells? Suppose what we can do in a chemostat, if you find out the Dmax value, then you can find out the what is the S value. S value is what? S is equal to what? I can write Ks Dmax mu max minus Dmax. I can easily find out this value. We can, and similarly I can find out the value of x. This is the x equal to this is the maximum value of x. The x max will be equal to what? Y x by s into S0 minus s. So this is the now x max. What is the what I can rate of growth of the cells, rate of production of cell is maximum when x into d, the x max into d max. If you multiply that, we can. So x max we have come across like this. Now we can do the calculation in other way also. Suppose we calculated the cell mass production. This is x max equal to y x by s. This is the S0 minus this one, that is the substrate concentration. That is the rate of substrate that is consumed, that you can find out the x max value. From that we can calculate this one. And finally we come across this x max value is this one, and at d max finally this equation, we derive this equation equal to x max equal to y x by s, S0 root over ks into plus S0 plus ks. The maximum cell productivity can be determined by the equation d equal to d max. Now you multiply it by the d with the d max, we will get this equation. This is equal to this is the maximum rate of production will be y x by s into d max into this, that what we have in the previous equation, that we can write in the 15 equation. See, in the 15 equation, this equation we can put this value, we can put this value of d max, d equal to d max, we can have this equation, this we can write this, we can find out the maximum cell mass production. Now in case of chemostat process, major drawback that we have the d washout, because the d washout, if you look at, I can show it like this, that if you have x into d related with the correlated with this one, this is like this. So you have two points. We have here you have d max value, here you have d washout value, d washout. Now at d washout value, I showed you before that here I have showed you that at d washout value this x will be 0, and if x will be 0, this corresponding point will be S0. That is the corresponding point will be S0. So if it is the S0, then what will be the d washout value? D washout value, d equal to mu under steady state condition, I can write mu max, S will be S0 plus Ks plus S0. So since there is no cell where no reaction take place, so S we can consider as S0, and if you put the value of S0, we can easily calculate that u. So we have both the expression with us, the d max value, and they are very close to each other. And in the industry, here I want to mention that in the industry, when you operate a chemostat process, that we don't have different pumps for the operation of different flow rate. The same pump we use for maintaining the different flow rate. And how we maintain the different flow rate? Just opening the valve, because we have seen in our wash basin, when you open your valve, then you will more water flow take place. If you close the valve, you will find less water flow. So you can control the flow of water with the help of valve. Now in the industry, that flow rate is controlling with the help of valve. Now by mistake, if the operator, then this is usually done by the operator, he increase the valve little bit more, there is every possibility that the Dmax will switch over to the D-washout. That is the major drawback that we have with the chemostat process. And now question comes that what is the physical significance of the D-washout? Why the D-washout takes place? The reason is that one reason I can tell you that I talk about the generation time. What is the generation time? The time required for the cell division. Now 1 by D is what? 1 by D is the hydraulic retention time. Hydraulic retention time is how long you allow the liquid in the reside in the reactor. Now suppose the cell has some generation time. Suppose this is a particular bacterial cell, usually they have the generation time might be 15 to 20 minutes. Now if you allow the liquid to reside less than 15 minutes or maybe 10 minutes, then what will happen? You will not allow the bacteria to grow. Before it grow, you are taking out the bacteria. So you are not getting any cell in the reactor at this D-washout. Now another way I can explain the situation. This is suppose this is a chemostat process. Now if we operate the chemostat process like this, now here I told you that there will be always in the major drawback of the chemostat process is that always there is a cell mass loss from the reactor. Now if your rate of cell mass that is going out of the reactor is more as compared to rate of growth of the cell, then what will happen? A time will come, there will be no cell present in the reactor. That is also the reason why we come across the D-washout in this reactor. So we should be very careful. We mention here as the D increases, S also increases slowly at first, then more rapidly as the D approaches mu max, corresponding X decreases, X tends to 0. So this D-washout is equal to and the correlation between the D max, D washout and D mu max is like this. D max is less than D washout and D washout is less or equal to mu max. Now the schematic that we can graphically I can explain like this. This is Dx, that is like this, and this is the typical one particular fermentation process that we have tried to find out this. This is where mu max value is 0.2. So here you see that your D max is around this 0.15, and where D washout is less than 0.2. You can see that it is not exactly 0.2, it is less than 0.2. So it is usually the D max is higher than D washout value. So you can easily find out, and they are very close, 0.15 and this is about less than 0.2. So this is very close. So even little increase in flow rate, there is every possibility we can meet the situation. Now this how we can find out that this kinetic constant of this K mu max and Ks we can determine in the continuous process also. If we plot 1 by D versus 1 by S, we can have this line of a buck plot, and from the slope we can get the value of Ks by mu max, and intercept we can find out the. So this constant value we can find out both by operating the chemostat as well as the batch process. So the drawbacks of the batch process, as I mentioned, that mainly the washout problem that we have, and how it can be safeguarded. It can be safeguarded by two ways. Either you recycle the cell mass to the reactor, whatever the excess cell that is going out of the reactor, you recycle back the same cell to the reactor, and otherwise you immobilize cell on the solid matrix so that no cell washout from the reactor take place. Now, let me discuss then that how you can analyze the chemostat with cell mass recycling, because the major drawback that we have with the chemostat can be overcome by recycling, and this is adopted by the different industry. I can give the example of activated sludge process. Activated sludge process is largely used by the industry for wastewater treatment process, because this is easily operated, and here this is the activated sludge process is nothing but CSTR with cell mass recycling. The cell mass recycling increase the rate of conversion, increase the stability and dampening the perception. This is like this. So we can write the balance equation is the input, cell generation, output, cell death we can assume to be 0 and accumulation. This is the equation I can write, and where alpha is the recycle ratio. Recycle ratio means I told you that by what you do that here the whatever cell mass is going out, we pass through cell separator. A part of the cell you recycle back to the reactor, and part of the cell you going out of the reactor, what you call cell mass wasting from the reactor. And here we can take out the supernatant. Here another way we can take out the supernatant. So effort that recycle, if the flow rate, this is the recycle flow rate, and in that inflow flow rate is F. Effort by F is considered as the alpha, what is considered as the recycle ratio, and XR is the settled cell mass concentration. This is the cell separator. You separate the cell. How you easiest way of cell separation is the sedimentation. Another way is by centrifugation. Also, you can separate the cell. So you will get the concentrated cell, and this is concentration with XR, and XR by X1 that is going out here. This is output cell mass concentration. We can assume to be C. Now, obviously, the XR and X1, this should be always greater than 1, because under no circumstances it is less than 1, because XR is the settled cell mass concentration, and this is the suspended cell mass concentration. Now, and we write this balance equation, we try to analyze this equation, and we come across the equation mu equal to 1 plus alpha 1 minus C into D. Now, this is very important, because C, as I told you, C value always greater than 1. If it is greater than 1, then what will happen? This will be negative, and this is the recycling ratio, and this if 1 minus negative will be less than 1, and so D will be mu divided by fraction. That means it can be operated at a higher growth rate, that can be operated. It implies that chemostat can be operated at the dilution higher than the specific growth rate of the cells. So because I can show you that in case of sterile feed and steady state conditions, that mu equal to D. I have already shown you. Now here mu is greater than mu. The D is greater than mu. So we can operate that. Your cell will not be washed out from the system. We can operate that from this. That is why we have written here a chemostat can be operated at dilution rate higher than the specific growth rate when the cells recycling is used. So again, this can be calculated in other way. This is the phase feed F into S0. This is the recycle feed. This is the outgoing feed that is going out. This is the material analysis, and this is the substrate consumption. This is on the basis of substrate balance. We can also write this equation like this. Now finally, we come across this x1 equal to this value. The x1 equal value can be again, therefore the steady state condition, the chemostat with increase by factor, the cell mass factor is this one. That we can increase the cell mass concentration by a factor of this. This is the substrate concentration of the effluent is determined by the equation from Monod equation, can be expressed by this equation. The chemostat with recycling is further analyzed, and we find out the value of S like this, and X1 we can express similarly like this, and the cell concentration and productivities are higher with the cell recycling, resulting the higher rate of substrate consumption. Now here that interesting thing is that here we find that X is increases. Now here that you see that in case of cell mass without recycling, that X value, this is the dilution rate. The here we get 1 might be the that C, approximately this is equal to cell washout. But when you do the recycling, this is shifting to the 2. That means you can operate it much higher dilution rate, because that is the advantage of cell mass recycling, that if you increase the dilution rate, increase the if you do the recycling, the dilution, operating dilution can be increased to a great extent. Now I want to point out that how to operate the multi-stage the chemostat in a series, because what you call cascade type of fermenter. Cascade, because you can remember that in case of reaction kinetics, we have come across this plug flow reactor. In case of plug flow reactor, we have come across this equation, this minus dCA equal to minus RA. Now here I try to point out that minus RA, if you plot by CA, and if you have this kind of thing, and this is the as the CA value decreases, this is CA0 to CA, 1 by RA increases, that means RA decreases. That is actually take place in case of product inhibition, because as the substrate concentration decreases, the inhibition occurs. That means the rate of reaction decreases, means inhibition occurs, and at this situation plug flow reactor is mandatory. Now we observe this plug flow reactor is very difficult to operate, because plug flow reactor there should not be any kind of back mixing. Now if you have in place of that, if you have multiple CSTR in series, then that will be equivalent to plug flow reactor 1, 2, 3, 4. So this is exactly we try to find out for secondary metabolite production, that growth and product formation step need to be separated, optimal each step and multi-stage of the chemostat process. So we can, not only that, we can have, because I can tell you this is very interesting, that in the anaerobic digestion process we have two type of culture. One is called acidogens and another is methanogens. The acidogens is the fast growing, if you as compared to methanogens. So if this is available, if you want to separate the acidogens from methanogens, you can do it very easily, because you have this picture is like this. One is this, another is this. So this is for your methanogens, and this is for acidogens. So if you here this is D washout for methanogens, and this is D washout for that acidogens. So if you use the dilution rate is very higher, then this organism, that methanogens, will be totally washed out from the mixed culture. So this is another advantage of this different with the help of dilution rate we can do this. Now in case of multi-stage analysis, we usually assume the steady state operation, no substrate addition for conjugate state, and balanced growth occurred. Now this is like this. This is substrate is coming, again whatever substrate is unconverted, we put it in the second reactor, so then more substrate will. But for the first reactor, we don't have analysis, is not problem, because we assume it is the sterile feed. We have already done, we have easily find out the value of S1 and X1 in this reactor. Now when you go to the second reactor, we shall have to do the material analysis, we shall have to do the substrate balance, and we shall have to find out what is the X1 and X2 value that we have. We can calculate S2 and X2 value in the second reactor, we can calculate separately. Then if we want to go to the third reactor, and we have to do the similar type of analysis. Like this, you can do the cascade type of analysis of the different reactor, find out the different cell mass concentration and different type of reactor. So let me stop here today, because we shall continue this lecture, and in the next class I shall discuss some problem also, so that whatever things I discuss, this will be little bit clear to you. Now in this lecture, I try to point out that the major drawback of chemostat process is cell mass that is that washout, and this can be overcome by the recycling. And by through recycling, is it possible not only to operate at a higher dilution rate, but at a higher dilution rate we increase the cell mass concentration to a great extent. The major concept of the cell de-washout I try to explain that d 1 by d is considered as the hydraulic detection time. If we have hydraulic detention time less than the cell division time required for the generation time, then say you are not allowing the cell to grow. That is why cell washout take place. Or we can explain in other way, if a cell mass that is going out of the reactor is more as compared to cell mass that is going in the system, if we allow the system to continue for longer period of time, we always meet the situation of de-washout. So this we can easily safeguard by using the recycling of the cell. And also we discussed in the last previous classes the immobilization of the whole cell. Through immobilization of whole cell also we can hold the cell in the solid matrix so that no washout of the cell will take place. So we discussed. I think in the coming lecture we try to discuss some comparison of chemostat and plug flow reactor. Then I shall discuss about this some problems, so that your how this can be applied for solving some kind of fermentation in the problem of the fermentation industry. Thank you very much.